The next part of the notes for Unit 11 is about bacteria, which are covered in Chapter 16. Bacteria, remember, are prokaryotes, and there are two kingdoms, two domains of bacteria, the archaebacteria and the eubacteria. We're going to focus more on the eubacteria in this unit, because these are the ones that we have more contact with. Basically, you need to remember that prokaryotes are definitely smaller than eukaryotic cells. They're from 1 to 5 micrometers in diameter and range from 10 to 100 micrometers in diameter, so they're pretty small. What you may not realize, because they're so small and because we can't see them, that they actually make up a large portion of the biomass in the world. It says in your book that the collective biomass of prokaryotes is at least 10 times that of all eukaryotes. So think about all the eukaryotes, including the blue whales and all of the humans and all of the other kinds of animals and plants and so forth, and realize that the prokaryotes make up 10 times as much mass. A large portion of your own personal mass is made of the prokaryotes that live in and on your body. This is the point of a pin, and the purple is the pin, and it looks kind of rough there, even though pins are very sharp. But you can see, because of the magnification, that it's not really as smooth as we have a tendency to think about a safety pin or a straight pin being. And the little orange things on there are bacteria on the point of the pin. So you can see they're very, very small, and you definitely cannot see them with the unaided eye. They live in lots of different places. They live in habitats that are very salty, very hot, cold, acidic. Okay, some bacteria are pathogens that cause disease, but by and large, most bacteria are benign or beneficial. We generally say about 2% of the bacteria are pathogenic, causing diseases, while the other 98% are either benign or beneficial. You have several hundred species of bacteria living in and on your body. They're involved in decomposing your dead skin cells. Some of them, the ones that live in your gut, help supply essential vitamins. And there are a number of them both on the surface of your body and inside your body that also help guard against against pathogenic organisms. Prokaryotes, one of the big things that prokaryotes do for us worldwide is that they help decompose dead organisms. So that helps recycle chemical nutrients in the ecosystem, and it's a very, very important job. There are 3 common cell shapes that you find in prokaryotes. The cocci are the spherical ones. These are round ones. Okay. Sometimes they occur in chains. These are called streptococci, and yes, that's where strep gets its name. Some are in clusters called staphylo, which is where staph gets its name. Bacilli are rod-shaped prokaryotes, and they can also be thread-like or filamentous. And then the spiral prokaryotes are like a corkscrew. The ones that are short and rigid, as well as being spiraled, are called spirilla, and the ones that are longer and more flexible, they're called spirochetes. So here we have cocci. You can see this is a streptococcus because it's chains of the cocci. These are bacilli, okay, and those are elongated, rod-shaped, and then here's a spirochete that is longer and filamentous. Now, one of the things that you need to realize about bacteria is that they do have cell walls. Remember, we talked about the fact that the ones that are eubacteria have a substance called peptidoglycan in their cell walls. All prokaryotes, or nearly all prokaryotes, have a cell wall. There may be a few that don't, but the cell walls are really important in maintaining the cell. When you stain eubacteria with a gram stain, the cell walls of the bacteria show up as being either gram-positive or gram-negative. The gram-positive ones have a simpler cell wall containing the peptidoglycan and large amounts of peptidoglycan, and they stain purple under the gram staining process. The gram-negative bacteria have less peptidoglycan and also a more complex cell wall, and these are the ones that are more likely to cause disease, and they stain pink under the gram staining process. This shows you the generalized bacterial cell, and you can see there this one has a capsule or a slime layer surrounding the cell wall, and there's also a plasma membrane or cell membrane inside the cell wall. You've got the fimbriae or the pili that are there on the surface that are for attachment. This particular one has a flagellum, which is for movement, and you can see the different things there. Inside the DNA or the chromosome is not in a nucleus, it's just in the central part of the cell. We call it the nucleoid region. It does have its own ribosomes, and it does have another secondary piece of DNA called a plasmid that is present in most bacteria. So various structures that you find there in the cell, but very, very simple. No organelles. Most don't have any internal membranes, although some may, but there's not really any organelles as such. They're about the size of mitochondria, so they don't have mitochondria. This shows you what the gram-positive and gram-negative bacteria look like on a stain. You can see the purple ones there are the gram-positive ones. So they've got the thicker, simpler membrane, the thicker peptidoglycan layer, and then the gram-negative ones have a thinner layer of peptidoglycan, and they're often ones that cause diseases. Both gram-positive and gram-negative bacteria cause diseases, depending on what the species is. The cell of a lot of prokaryotes is covered by a capsule, which is a sticky layer of polysaccharides or protein, and that enables the bacteria to stick to a substrate, to a surface, or to other individuals in a colony. It also helps protect them from attacks by the immune system of the host, because it's another layer that you have to get through. Here you see a bacterium capsule stuck to the surface of a tonsil cell, so it makes it easier for them to cause the infection because they can adhere to the surface while they're secreting their toxins or other substances. So some prokaryotes have these other structures, like we said in the diagram. Some have flagella, which will help them move in their environment. Some have these hair-like projections called fimbriae or pili that help that allow them to stick to each other or to their substrate, similar to the way that the capsule may help. The pili or fimbriae are more like Velcro, basically, that gives them an attachment. Here we see some fimbriae. These are longer fimbriae on a flagellated bacterium. Sometimes the pili are located in the same arrangement, but they're smaller. Prokaryote population growth occurs by binary fission. They don't undergo mitosis. They only have one chromosome, which is a circular chromosome. And so it's much easier and faster for them to reproduce, and they don't have to undergo the complex processes and stages of mitosis like eukaryotic cells do. They can rapidly produce a new generation within hours. A lot of bacteria replicate every 20 minutes or so. They can also have a lot of genetic variation by spontaneous mutations that can occur, and what that can do is guarantee that some members of the population will survive whatever the changes are in their environment, whether it's an antibiotic or whether it is different nutrients and so forth. So they can change pretty quickly, evolve pretty quickly, because they reproduce so often. The genome of a prokaryote has about 1,000th as much DNA as a eukaryote. So it's one circular chromosome backed into that nucleoid region of the cell. And many of them, if not most of them, also have additional DNA molecules called plasmids that are smaller circular DNA molecules, and they're independent of the chromosome and may be passed from one bacterium to another. Here we see the chromosome. This is a bacterial cell that has been disrupted and ejected its nucleic, its DNA. And so you see the chromosome is this large area of all these swirls here, and the plasmids are separate small pieces that you see there, separate from the main chromosome of the cell. Some prokaryotes are also able to form these specialized cells called endospores. Endospores can remain dormant through harsh conditions. They can survive extreme heat or extreme cold. They can survive not having any water. All different kinds of things. You can see here the endospore inside the bacterium. You've got the genome there, and it's surrounded by several layers of cell wall that are protecting it from drying out or being exposed to harsh conditions. And this is a good thing for the bacteria, not necessarily so good for the host that can come in contact with it later and reactivate it. In terms of reproduction, of course, the type of reproduction that the bacteria undergoes for growth is called binary fission. This is asexual cell division. It's just making new copies of the cell. It's kind of like a cloning process. There's also a couple of ways that bacteria can exchange genetic information or recombine genes, and we'll talk about each one of these in turn. In transformation, this is when bacteria pick up DNA from another source. When we talked about the discovery of DNA and learning that DNA was the molecule of heredity, we talked about the fact that scientists had put dead pathogenic bacteria with living non-pathogenic ones, and that made the living ones become pathogens. That's called transformation because they pick up DNA from another source. In conjugation, living bacteria can exchange genetic information through means of a tube that they can grow between cells and pass a copy of their plasmid into the other cell. This is how a lot of bacteria transmit resistance to antibiotics within their colony. Transduction is when you have genes that are transported by bacteriophage. If the phage picks up or some of the bacterial genes get included with the viral genes in the making of the virus, then the bacteria that get infected with the phage can then pick up genes from the phage from different bacteria. And then, of course, the last one is the endospores, the resistant cells that can withstand harsh conditions. Here's how binary fission works. We've got our DNA that gets replicated there. Notice that each of the chromosomes in the nuclear area are the nucleosome region are attached to the cell wall. As the cell grows, the cell membrane pulls those two apart, and then the cell wall and plasma membrane start to divide, forming cross walls in between the two cells, and then eventually the cells can separate. And this kind of shows you in diagrams, and then here's a micrograph showing you one that is starting to divide there, the partially formed cross wall between the two nuclear areas containing DNA. In transformation, of course, here we have a donor cell donating DNA to a recipient cell, and it picks up those genes. Transduction is when you've got a phage-infected donor cell that transfers bacterial genes to the recipient cell, and then conjugation, where they actually exchange plasmids with each other, transmitting some genes to the other cell. Prokaryotes have a lot of nutritional diversity than eukaryotes. There are two main kinds of energy that can be used, of course. Phototrophs capture energy from sunlight, and then chemotrophs use energy stored in chemicals. And so we have to talk about how that affects and where they get their carbon from as well. Two sources of carbon are used by prokaryotes. Autotrophs get carbon atoms from carbon dioxide, like plants do, and heterotrophs get their carbon atoms from the organic compounds in other organisms that they can infect. The terms, now what we do is we actually combine those, talking about how they get their energy and their carbon source. And this describes the four modes of nutrition in bacteria. Photoautotrophs get energy from sunlight and use carbon dioxide for their carbon source. Photoheterotrophs get energy from sunlight but get their carbon atoms from organic molecules. Chemoautotrophs harvest energy from inorganic chemicals and use carbon dioxide for their carbon. Chemoheterotrophs get their energy and carbon from organic molecules. So there's very complex things going on there in the bacterial world. Here we have some examples. These pictures kind of in a graph where you talk about their energy source across the top and their carbon source down the side. And these are just some examples of ones that perform these different nutritional modes. In addition to their different carbon sources and energy sources, bacteria also have differing abilities to handle oxygen. Those that are what we call obligate aerobes require free oxygen. They cannot live in the absence of oxygen. Facultative anaerobes can use oxygen if it's present But if it's not present, they can use fermentation to produce energy. And then finally, the obligate anaerobes are poisoned by oxygen. So they can only use an anaerobic respiration process like fermentation.